Hey class, welcome to section 2.5 lecture through YouTube. And so if you have your DQ out, I'll let you answer it so that way we can get a little bit closer to rolling the dice for some prizes. So take a look at this graph. So yesterday you had to draw two graphs showing endothermic and exothermic reactions. So take a look at it. See if you can figure it out if it's endothermic or exothermic. Maybe take a guess. Try to remember what you learned yesterday and then look back at your notes. Okay, so if we do look back at our notes, boom, energy absorbed. Okay, so this is an endothermic reaction where the reactants have a lower energy level than the products. And this is the energy absorbed in that process. Endothermic reactions. Okay, moving on to section 2.5, which is probably my favorite of the chapter. We're going to look at enzymes. And so an enzyme that's used in biological chemical reactions, we usually call enzymes catalysts because they help start the reaction. So you may have used the term catalyst maybe in a paper, just something that starts a process or an event. And then we're going to talk about how enzymes help regulate some of those chemical reactions. So a little bit of a preview here, just to kind of get you thinking. If you leave a newspaper in sunlight, it will eventually turn yellow. My husband, he gets the Wall Street Journal every single day. And so I'll glance at it, page through it, and then I'll stick it in a box. And this box sits right by our porch window. And you can kind of see this gradient of super yellow at the bottom to newer white newspaper at the top. So what's going on there is a chemical reaction is actually taking place. Now, if you set a match to the newspaper, it's going to burn quickly, okay? And so what does adding energy to a reaction do? Okay, it's going to cause a chemical reaction, like the reactants will undergo a change that was different before. So the products will be completely different. Here with yellow paper, here ash. So it's increasing the kinetic energy of the reactant molecules, and as a result, you're going to see more collisions occur. And when collisions occur faster, in a faster rate, then you're just going to get a faster reaction. And this is another reason why I like this section. I usually bring in the newspapers and light them on fire. And now I'm at M states. So, all right, moving on. So 2.5 enzymes. So, feel free to just pause the notes right now, write down the slide, and then listen to me talk because I'm going to go through this pretty quickly. I mean, this video is probably going to be about 10-15 minutes long. So anytime you have to pause to write, and then you can just listen afterwards. So a catalyst lowers activation energy. And so yesterday, activation energy was a vocab term. Activation energy is the energy required to start a chemical reaction. So we talked a little bit yesterday about the scenario of biking up the hill. It'd be the energy of getting to the top of that hill. So a catalyst decreases this activation energy, and as a result, it's going to increase the rate of chemical reaction because it's not going to need as much energy. Okay, so it's kind of like taking a hill and cutting it in half. Not as much energy, and then you could still undergo the chemical reaction. Now, the thing I want to stress with catalysts or enzymes in a chemical reaction is that they take part in the reaction, but they're not reactants or products. A lot of times you don't see catalysts listed in the chemical equation or the chemical formula. And there's a couple of reasons for that. First off, reactions can go forward or backwards using the same catalyst. But another reason, what was I going to say? Is that the catalyst is actually reusable. Okay, so down here I have this simplified diagram of a catalyst, and you can see that it's very specific to what it's going to bind to. So in this case, it's going to bind to reactants, and it will undergo a chemical reaction of some sort. In this case, we're going to bond purple to orange, and you can see that they are held more closely together. And then once that chemical reaction takes place and a new bond is formed, or maybe it's released, depending on what it does, it releases the new products and it goes to find more reactants to repeat the process. So they are not used up or changed. The catalyst remains the same shape as it did before and after. So they're not reactants or products, they're not used up, they are reusable. You don't see them in chemical formulas or equations. So I showed you a graph that was similar to this yesterday with if you don't have a catalyst or an enzyme, well, geez, you got to really climb that hill. And what catalysts and enzymes do is they lower the amount of energy needed. Okay, so here I have, down here it says nitrogen and hydrogen equals ammonia, and this is the amount added. But if I have a catalyst, if the catalyst goes around and finds nitrogen and hydrogen faster, and matches them up. I said yesterday it's kind of like a matchmaker, and will force them to get together to form ammonia. If you don't have catalysts, well, now it's kind of left up to fate and kinetic motion. So, you could increase the temperature because increasing the temperature will increase the kinetic movement of these atoms, and then forceful collisions will cause a chemical reaction to occur. But if it's just, room temp, it's going to be a pretty slow rate. It's going to be a really boring thing to watch. So that's why we use catalysts. So a diagram to kind of get, again, to show the difference between having an enzyme and not having an enzyme. So imagine we have some derived frogs here. Those are the reactants. And the products is to get wet frogs, okay? And we have a high energy barrier. This frog needs to leap extremely high to become a wet frog, okay? So it proceeds at a slow rate if at all, But if I were to, take out this barrier, lower it in some way, so that's kind of like using an enzyme, decreasing the activation energy needed. Well, now that reaction is going to happen sooner or at a faster rate because not that much energy is needed, okay? It's a lot easier to jump over a pile of rocks for this frog, so to speak. So enzymes are catalysts for chemical reactions in living things. Okay, so they jumpstart chemical reactions. They lower that activation energy level, so important things can happen. So it lowers activation energy, increases the rate of reaction because it binds to the reactants. Okay, and then forces the chemical reaction to take place. Causes that collision, matchmaker. Reaction of this is reversible. Okay. Now, enzymes are also proteins, and this is extremely important because proteins are really sensitive to their environment. If you take college biology next year, you're going to learn that proteins actually have 4 levels of structure. And the first one is the amino acid sequence. So in chapter 2.3 or section 2.3, we discussed that there are 4 large macromolecules of life: nucleic acids, lipids, carbohydrates, and proteins. And then we talked about their monomers. Nucleic acids, the monomers are nucleotides. Lipids, for the sake of simplicity, we're going to say fatty acids are the monomers. Carbohydrates, monosaccharides, or simple sugars like glucose. Now proteins, the monomer is amino acids. So amino acids, the sequence of that is actually the first level of structure in a protein. And the sequence of the amino acid basically determines what kind of protein it's going to turn out to be. So I also gave you the example of hemoglobin, how it looks like a nice little donut-shaped red blood cell, right? And if one mutation causes a different amino acid, well, it's no longer donut-shaped, it becomes sickle cell shape and renders the function of it almost completely useless, okay? So, well, that's the first level, sequence of amino acids. Now, the second level has to do with how these amino acids interact with each other as well as the environment, because some regions are polar, some regions are non-polar, so they tend to form different, cross bonds, if you will, and kind of alters the shape of it, ties into tertiary with ionic bonds and covalent bonds and disulfide bonds and all that jazz. But the structure of the protein is dependent on, whether or not it's going to function properly. So the environment can alter this structure. So if you change the structure, you might ultimately change what it does. Will it work right? Will it work as efficient? So a couple different ways you can change the environment. Temperature, by either increasing it or decreasing it. Or you can change the pH, make it more acidic or more basic or maybe even more neutral. So what happens if I make this more acidic? Well, now maybe it's not going to be too, round shaped here for the enzymes. So then these guys can't bond properly anymore. Okay, so it changes the shape of how reactants bind to it. I also told you the story about my college research. I know I keep bringing this up. With PP5, the shape of the gummy bear, and we were trying to mutate parts of the substrate to see if it will still bind to the gummy bear arms. So, that was another example of structures tied to function. And if you change that, it may not work right anymore. Okay, so a substrate is a specific reactant that enzymes act on. So these are my enzymes. They're the proteins that, the matchmakers. And these are substrates, the things that bind to the enzymes. And they are very shape specific, we'll say. So you can obviously just look at the two different shapes and say, okay, green and red, they're going to bind to this enzyme. And over here, this magenta fuchsia color is going to bind down here, and blue. So these are my reactants, okay, also known as substrates that will bind to it. And then a chemical reaction will occur, and then you get products, and they will get released. So we call the parts on the enzymes where substrates bind to active sites, because that's where all the activities taking place. So you can think of active sites as the locks, okay, the shapes of the keyhole, if you will, and the things that bind to the enzymes as keys, because they are very specific. So like in your mouth, you have salivary amylase that, it's found in your saliva, and it breaks down complex carbs into simple carbs. So it kind of starts the process of breaking carbs down into monosaccharides. So active sites, locks, and then substrates, keys. So down here I have the enzyme, here's the substrate, this is the active site. Very shape specific. Substrate binds to it. I have formed a complex here. Chemical reaction occurs. In this case, we're going to break the substrate down, kind of like what salivary amylase does with complex carbs, breaking it down into maybe monosaccharides, and these are my new products. So here's glucose substrate in a bind-in site that binds to glucose, and you can see that it kind of just nestles right in there perfectly. Nom, nom. Okay. Another thing I should probably stress that I really like about this diagram is that when the substrate binds to an enzyme, the enzyme usually undergoes a shape conformation, or like a shape change, if you will, for a little bit while the chemical reaction takes place, and then we'll open back up and release the products usually. Okay, so there's 4 steps in the process of how an enzyme works. Okay, that top part doesn't even make sense. 4 steps in the process of an enzyme working? Okay, whatever. All right, so step number 1, enzyme and substrates are in the same area. Okay, step number 2, the substrates binds to the active site on the enzyme. So come in and bind to it. Step 3, substrates are changed. Maybe they're broken down, or maybe they're combined with the other molecule, like in this picture down here, to become a new product. And then the enzyme lets it go, and it goes to find more substrates to carry out the chemical reaction. Okay, so that is 2.5 in a nutshell. I'm going to talk a little bit about the liver lab tomorrow. So in your liver lab tomorrow, we're going to look at beef liver, and inside beef liver is an enzyme called catalase that breaks down H2O2, a very deadly substance, into water and oxygen gas. In this experiment, you will be changing the pH. So I'm going to have some different pH levels, 1, 3, I'm hoping to get a 5, a 7, a 9, 11, and we can't really measure the 14 because our pH strip only goes to 12. But we're going to alter the pH of it. And then we're going to figure out which pH level is most optimal for the catalase. So you're going to think about, well, geez, liver is found in this case, a cow. What is the pH level inside of a cow that allows it to, work at its optimal best, breaking down this very dangerous substance into harmless substances, if you will. So, yeah, that's the experiment. How you will determine that will be with foam height. And so foam height means that the higher the foam, the more, the higher the reaction rate, we'll say. Basically, it's more optimal there. So you're going to be measuring foam height in your experiment. On this top diagram looks at pepsin, which is a enzyme found in your stomach. And you can see that it works best at the pH of 2 and not so much at pH 4 or even extremely acidic 0. So, even things that thrive in acidic environments have an optimal pH. Same thing with trypsin, pH of 8. So, okay, that does it. Thanks for watching. You do have a 2.5 worksheet. So work on that and then be prepared tomorrow for the liver lab. All right, take care.